In this video, I'm going to be covering a 6.1.1 of the European Baccalaureate Biology syllabus called Cell Structure. So as you can see here from the screenshot of the syllabus I took, we are going to be covering the eukaryotic cells and organelles and how to draw them and label them. We're going to compare these cells under different microscopes, and we're going to be able to differentiate these by understanding the relative scale. And over at the end, I'm going to talk about labeling mitochondria and chloroplasts. Before talking about and drawing the structure of eukaryotic cells, I want to talk about their organelles. Now, what are organelles, you might ask? Organelles are very simply what organs are to us as humans. So they are specialized parts of the cell which have specific functions they carry out. And there's a list of about 10 we have to go over for the syllabus. And I have a cheat sheet, you could say, down below in the description that I used to study, which has all the definitions that I kind of memorized and hopefully will help you study as well. So going over and explaining it, first there's the cell wall. Now the cell wall is only found in plant cells, which is why I'm adding a little green star here so you don't forget. And it provides structural support to the cell and it helps maintain the shape of the plant cell. Now, next up, we have the plasma membrane, which is in both animal and plant cells, and it controls what enters and exits the cell. So this provides the protection and the support, and it is involved in cell communication. Now, the plasma membrane is a topic we're going to go over later in the year in S6. And this definition is a very, like all of the other definitions, it is very superficial compared to what this organelle actually does. But we'll go over that in a later video. Next, the cytoplasm. So the cytoplasm is kind of the rest of the cell. It is a gel-like substance that fills the entirety of the cell, and it's what the organelles are inside of. And this allows for communication between the organelles. Next, there's a nucleus. The nucleus is arguably the most important part of the cell. This is what holds all of the genetic information, and it regulates gene expression. So the genetic information I was talking about is DNA. And inside the nucleus, there's the nucleolus, which is a region in the nucleus, which is even darker. And then you have the chromatin. So chromatin are kind of the strands of DNA, very simply. They're the strands of DNA that are in the nucleus, and you will see these in the diagrams I show you in the next kind of section. Next up, we have the vacuole. So this, I'm also going to put a star next to, but not a green star, but it is important to note that the vacuole is different in animal and plant cells. In animal cells, it is often smaller, but in plant cells, it really fills up the rest of the cell because it's what gives rigidity to the cell wall and is what enables the cell wall to be rigid and expanded fully. Now there's also mitochondria, which we will go more in depth on later in the video. They produce ATP, energy. This is very easy to memorize. And everyone kind of memorizes the definition. The mitochondria are the powerhouse of the cell. So they are what produces the energy. Next up, we have the rough endoplasmic reticulum, which I will just write as RER in the video. But down below in the description, again, there's the cheat sheet. And this is involved in the synthesis and the sorting and transporting of proteins. Unlike the SER, so the smooth endoplasmic reticulum, which takes part in the synthesis of lipids. So here I'm just going to very quickly put proteins here. And here I'm going to add lipids. Now, next up, we have ribosomes. Ribosomes are on the surface of the rough endoplasmic reticulum, which is why the rough endoplasmic reticulum is rough. And these translate the mRNA into polypeptides, as you might remember from S5 when we covered protein synthesis. The two last organelles are the Golgi apparatus, which modifies, sorts, and packages proteins. So this is kind of one of the final steps in protein synthesis, takes place in the Golgi apparatus. Sorry, let me write that a little bit neater. And chloroplasts. So chloroplasts are the organelles in which photosynthesis occurs. Now, again, important to note, these are only present in plant cells. So as you can see here from the 2 green stars I wrote, boom, remember, these are only present in plant cells. Hopefully you can read this handwriting. It's hard to draw on this tablet, but I'm going to hopefully get better at it over the course of these videos. So here are, again, all the organelles. I have mentioned all of their definitions, and down below I will have linked for free a cheat sheet with all of the definitions that you need to know. These are all the definitions that I memorized, and it worked fine for me in the exam. What's important is you understanding their role in the cell, and being able to identify them in pictures of the cell. So that's what we're going to move on to right now. Okay, so now we're going to talk about the structure of eukaryotic cells. So if you're wondering exactly what a eukaryotic cell is, a eukaryotic cell is a cell where there's an obvious nucleus. Now, in both of these, we can see a pretty evident nucleus, which is this big bubble right here, with a darker spot in the middle. Now, in this image, you can see very clearly what the normal cell is. So here, this is the cell, a full cell. It's an animal cell. And they've shown the cytoplasm without the organelles inside of it. And here you can see this nucleus, which is the little round thing in the middle, which is pretty obvious, and the nucleolus inside of that nucleus. Now, zooming in closer, we can also see the chromatin inside of the nucleus, and the chromatin is what holds the genetic information. So the chromatin is essentially just the DNA which has been twirled up. Now, this is one of the pieces of information on the syllabus which I had a lot of struggle finding online, and this picture really helps understand where everything is. Now, moving back to the rest of the organelles in the cell, which is what we're going to be placing in this section, and this is what helps you understand the structure of the cells. We're going to go over all of the organelles and where they're located. So starting off, we talked about the nuclei. This is a nucleus, and this is the nucleus of a plant cell. There's the nucleolus in the middle, and there's also chromatin. Now there's some chromatin is missing on the actual plant cell here, so I'll just add some very quickly, just these kind of squiggly strands. Now there is 2 key differences between plant and animal cells, 3 actually. One is that the plant cell is rectangular, as you can see here in the simplified diagram. It's more rectangular, and the animal cell is more circular. Another key difference, which I'm going to go over straight away, is the cell wall, which only the plant cell has. Here you can see this thing here is the cell wall, which only the plant has, whereas the animal cell only has a cell membrane. Now this is the plasma membrane or cell membrane, you can call it whatever you want. And the plant cell also has a cell membrane. Here it's this part, let me take a different color. It's this part, which is inside of the cell wall here. So yeah, that's one of the key differences. The plant cell has a cell wall, whereas the animal cell doesn't. And also the plant cell has mitochondria, which the animal cell, as you can see, doesn't have. Now, moving on to the similarities of both of these cells, let's go over the mitochondria. So the mitochondria are here in pink. This pink highlighter is not very obvious. Oh, there it is. It's fine. These are the mitochondria, powerhouse of the cell. That's a simple definition we memorized, but there's obviously a more in-depth definition to it. Powerhouse of the cell right here. Both of the eukaryotic cells have one. Next, we have the rough endoplasmic reticulum, which is on the outside of the nucleus. And it's rough. It's very easy to see that it's rough because it has these small bubbles on it. Hope you guys can see those clearly. And what those small balls are actually the ribosomes. So the ribosomes are, let me take a different color for this. The ribosomes are these small dots here that you can see inside of the cytoplasm of the cell. These small dots are the ribosomes, and they are on the edge of the rough endoplasmic reticulum, which is why it's called rough. Next, there's the smooth endoplasmic reticulum, which looks like the rough one, except it's not rough. It doesn't have any of the ribosomes on it. Here labeled on both the animal and plant cell. And quickly, I mentioned the cytoplasm, if you were wondering what exactly that is. It is the This is very simply all this white matter you see. These are the cytoplasm of the cell. It's what contains all of the other organelles. We have the Golgi apparatus, or Golgi apparatus. I don't know how you're supposed to pronounce it, but it is right here. The vacuole, which is also a key difference between plant cells and animal cells. The vacuole in plant cells is a lot bigger, and it exerts turgid pressure on the cell wall to make it so that the plant isn't flaccid. Here in the animal cell, you can see it's a lot smaller. And that's it. That covers all of the structure of the eukaryotic cells. I hope these diagrams have helped you understand better where all of these organelles are located within eukaryotic cells. And what I would recommend for studying this topic and this part of the syllabus would be simply practicing annotating these cells. So what I have in the description down below is my worksheet that I had made to study, which consists of these diagrams that are on the screen right now, with arrows leading out from the organelles that are important to annotate based on the syllabus, and that will hopefully enable you to practice memorizing where these organelles are, and you should also memorize their functions, which I went over just before this. Hopefully you have a good revision session, and I'll see you in the next part of this topic. Next, we're going to look at how cells appeared under different microscopes, and how knowing the relative sizes of organelles are going to help us identify these in micrographs. So here I have found a good picture of relative sizes, and I'm going to walk through it very quickly, and this will really help us with the next section. So at the very left, we start with atoms and small molecules, which you see right here, from 0.1 to 1 nanometer. Now moving up, we see the proteins and lipids from 1 nanometer to 10 nanometer, and then viruses at around 100 nanometers. Then we have the smallest bacteria, just a few nanometers across. Now, for what I had done to revise is I had only used, here, let's take this, I had only used, like cropped it up, and I had only used this part of this picture to study. This whole section with the electron micrograph and the light microscope is very important for the section I'm going to talk about next. So this is a very nice reference to have. So now we're going to move on to a light microscope. Now, light microscopes can reach a magnification of around 1000x, but the resolution is only about 200 nanometers. So this means that we can easily see cell boundaries, nuclei and vacuoles, but not smaller structures like ribosomes or fine details of the mitochondria. So here in this picture we can see what a light microscope looks like. You might recognize these. These you will use probably in class, and you probably have them in school. And I personally used these last year to look at some animal and plant cells. So these aren't pictures I took, there's some I found on Google. But here you can see an animal cell down here, under a light microscope. You can clearly see the circular structure, and you can see the darker nucleus in the center. You can see some darker regions, which are something inside, but we can't identify clearly what these darker spots are in the cell. Same with a plant cell. You can tell this is a plant cell by its kind of sharper edges and how closely packed together the cells are. Again, you can clearly see the nucleus, these rounder circles, but it's kind of hard to identify the other organelles. You can see some greenish elements, but it's hard to identify exactly what these are. This is where electron microscopes come in. They have a resolution down to 1 nanometer, which is over 200 times better than a light microscope. And this allows us to see organelles in much greater detail. So as you can see here in the animal cell and the plant cell micrograph, it is very easy to see clearly the different organelles inside of the cell. So first and foremost, you can see very clearly this region is the nucleus, and you can see the nucleolus right here, the darker region. In this animal cell, again, you can see very clearly the nucleus. You can't see exactly where the nucleolus is. I'm guessing it might be this, but I wouldn't say so for sure. but you can definitely recognize regions of spaces way more clear as well apart from the nucleus. You can notice the red of endoplasmic reticulum, the RER, the chloroplasts here, which I'm going to label. And all of this I found on a website, which I will show you right now. First, let me label the chloroplasts. So here, the chloroplasts are very clear. So let me show you the website I used. And it was very useful with studying how you can see the organelles in electron micrographs. That was a key part of my exam, so I would highly recommend doing so as well. Okay, so if I zoom in a little bit, this is the website I used and where I took these pictures from. This is very nice to be able to tell and locate all of the different organelles, not all of them, but a lot of the organelles in these micrographs. So here you can highlight it. So if you put no features, nothing will be highlighted. But if you highlight, for example, nucleus, as you can see, the nucleus is highlighted. If you want to highlight the chloroplast, like I showed you, the chloroplast is highlighted. The endoplasmic reticulums are highlighted only in the animal cell. There's some that you obviously can't see still, but it is still better than the light microscope. Then the mitochondria. It takes a little bit of time to load, but everything is there. And you can recognize these. Oh yeah, this makes sense. It's the Golgi apparatus. It's a bit curved, like we saw in the simplified drawings. The chloroplast we saw, the cell wall. Okay, this makes sense. I could have seen this. I could have noticed it myself, etc., etc. So this is very, very useful for studying. And then down here you can see a deeper, more in-depth, clearer picture of the actual organelles. So etc. This website is very good. I highly recommend going onto it. It's in my sheet in the description, and I will also paste this in the description very simply. Finally, let's zoom in on two of the most important organelles for energy, the mitochondrion and the chloroplasts. For the exam, you should be able to label them, so their structures, but you don't have to know the functions of these structures. So here's the mitochondrion, the powerhouse of the cell. It is where aerobic respiration happens and ATP is produced. Now, on the outside, we have the outer membrane. This is a smooth barrier. Sorry, the handwriting is not great. This is a smooth barrier that separates the mitochondrion from the cytoplasm. Here it is. This is a simplified drawing I found on Google. I don't remember exactly what the source is. I will have it in the document. But I had essentially to revise, just covered up the actual labels, and that's how I practiced. And that's what I have done also in the worksheet, so you guys can practice. Here we have the inner membrane. It is folded into cristae, so these are the cristae. And these folds increase, so the cristae increase the surface area for the proteins and the enzymes involved in respiration. Here we have the inner membrane. So because there's an outer and an inner membrane, there's an intermembrane space. This you don't need to know for the exam, and you don't need to memorize this, but you know, it's nice to know. The space inside all of this is called the matrix, not the movie matrix, just the mitochondrial matrix. It contains enzymes for the Krebs cycle as well as the DNA, which you can see here in a spindle, as well as the ribosomes. And that covers it for the mitochondrion. So because the mitochondria have their own DNA and ribosomes, they can make some of their own proteins, which supports the endosymbiotic theory, which is the idea that mitochondria originated as pre-living bacteria. This is a cool fact that I found and I thought would be interesting to share. You don't need to know this for the exam, however, I thought it would be good to share. Maybe it might end up being useful, you never know. Moving on to the chloroplasts. They are found only in plants and some algae. It is a site of photosynthesis, where light energy is converted into chemical energy in the form of glucose. You probably, again, already know this, however, it's still important to note it. So like mitochondria, chloroplasts have a double membrane. So here you have the outer membrane. the inner membrane, and the intermembrane space. The fluid interior is the stroma. It contains enzymes for the light-independent reactions, along with the DNA right here, and the ribosomes, which you can see here in orange. Within the stroma are thylakoids. These are flattened membrane sacs that contain chlorophyll and are the site of light-dependent reactions. And here we have lamella. Thylakoids are often stacked into graina, as you can see here. And this maximizes the efficiency of capturing the light. Now, together, these structures allow for the chloroplasts to absorb light, produce ATP and NADPH, and then use them to fix carbon dioxide into glucose. Now, you might have noticed I had crossed out two of the labels, the starch granule and the drops of lipids. This is because these are not indicated in the European Baccalaureate S6 syllabus. However, these are still interesting to know, which is why I only crossed them out so you can still see what's actually written. As a quick note, we need to remember that both mitochondria and chloroplasts have a double membrane. They both contain their own DNA and ribosomes, and they're both involved in energy conversion. Mitochondria release energy from glucose as ATP, while chloroplasts capture energy from sunlight to make glucose. Being able to label these diagrams is very, very important for the exam, and recalling their functions isn't, but it is still interesting and it is nice for me to tell you, so maybe you can remember it when we get to S7. This is it for the video. Hopefully it has helped you understand and cover all of S6.1.1. Now, if you're looking for the revision list and the cheat sheet I made, I have added the document in my Patreon. As you can see here, I'll show you how to navigate it. Into collections, S6 biology, you will see S6.1.1 cell structure. Here I will have pasted the YouTube link, and down here you have the PDF. Here are some basic guidelines for what the PDF includes, but here you have the PDF, and there's everything in it that you might use to revise. So supporting on Patreon is optional, but everything is completely free. I want that to be very important. I want it to be free for you to be able to learn. Thanks for watching and good luck with your revision.